Welcome back to another episode of the DNA of Things, where we explore the human potential at the crossroads between DNA, environment, and behavior. Here, we uncover how understanding our genetic code and its interaction with our surroundings, our lifestyle, can unlock transformative potential in health, personal development, and ultimately realizing our human potential. Now, today's guest, Chris Barbazette, is a very important person to allow for the unlocking of human potential at scale. Chris is a distinguished leader in biotechnology commercialization with over a decade of experience scaling life science companies like DNA Script, Result Biosciences, and Affymetrix. Now, Chris specializes in advancing groundbreaking molecular technologies in global markets, making innovative healthcare solutions more accessible. In this episode, we dive into Chris's fascinating career trajectory and his unique role in bridging scientific and commercial worlds to bring new technology into practice. We explore the global efforts to democratize access to diagnostic and genomic tools, insights on the international genomics market, and his process for transforming innovation into widely adopted applications. So this is personalization at scale. In this episode, you'll gain insight into the future of personalized medicine and why advancing global access to biotech is crucial for broader healthcare transformation. Let's dive in. Chris Barbazette, an accomplished commercial leader in the biotechnology or tech bio and life science sectors, has held pivotal roles at companies like DNA Script, Result Bioscience, and Affymetrix. Affymetrix acquired by Thermo Fisher. Chris is focused on bringing DNA and molecular techniques to market. He's currently a commercial advisor for Backstitch DNA and an executive in residence at General Inception, where he helps scientific entrepreneurs scale their innovations globally. Chris's expertise spans commercialization, market development, and global expansion strategies for diagnostic and life science companies. His recent focus includes high throughput DNA engineering and the international expansion of lupus testing. So, Chris Barbazette, welcome to the DNA of Things.
Thank you. Thanks for having me. Appreciate it.
Oh, my absolute pleasure. Well, for the benefit of the listeners, it's important context, I suppose, to share that, well, gosh, we've known each other, or at least known of each other for, I think it's about closing on 10 years since our Athletigen.
Probably about a day.
Yeah. It was our Athletigen Affymetrix partnership. So it was just like the very beginnings of the consumer genomics industry. And, I'm sure we're going to talk a little bit about that in general, but you are, a man of many talents in terms of bringing, the technologies for personalization, precision medicine, R&D, discovery, and biotech and tech bio, bringing those things to bear. And so what I'd really love to do is, we're certainly going to talk about some of your experiences in particular, but how did it all start, man? Like, how did you become this kind of, generalist as it relates to, precision medicine, let's say?
Yeah. So, I think any person, it's a long journey and it's not a straight line. So for me, actually, my first real full time activity was starting at Affymetrix, and Affymetrix definitely had its ups and downs, and I came in and I was at Affymetrix almost 10 years. And when I first came in, the company was struggling with how to make the technology it had relevant. It needed to iterate the technology, and so I actually came in from outside the industry. So it's one of the things that I think usually surprises people. There's a typical pathway I think that a lot of people take, and that's in the commercialization world or commercial world, that where you start as someone doing a PhD, and then you decide working in the lab's not for you, and you want to sell into the lab, maybe become an application scientist, and then move into sales, and then move up as you do well in sales. And that's absolutely not the path I took, and so that makes me, I think, a little bit unique. And so I actually, when I first started at Affymetrix, spent a lot of time learning, teaching, or teaching myself, and doing it through self-study, but also through understanding and working with some of my colleagues, and also doing some external classes, to the point where I wanted to be able to be fluent in being able to talk to people about the different technology that Affymetrix had, and being able to explain it, and being able to hold my own in a conversation. And so, I think that really having, and I think the value then that I was able to bring is that I had a perspective that was unique and outside the industry. I wasn't like everyone else. The other part is that I, early in my career and earlier in my life, I spent a lot of time in Asia, and I actually studied in Japan. I did an exchange program in Japan. So Affy at the time had some challenges and some turnover where they needed help in Asia, and I helped the company in Asia, and what was a 3-month temporary assignment ended up becoming a 6-year assignment that where I worked on genomics all over the world, and that I think really helped develop my career, but also inform my what I felt was important, and thinking about various stages of commercialization, how to commercialize things, but also how to think about the customers, and the different types of customers, and really trying to make things truly global. So I don't know if that answers your question, but that just kind of gives you hopefully a sense of how I started, and how I in what we do, and how it started to develop.
Yeah, no, I mean, that's a great overview, and maybe we can use Affymetrix as a case study here. I mean, because it certainly was in terms of orienting you to the market, but I think it's also a great conversational tool as it relates to genomics and the DNA industry. Where you've got. I mean, when was Affymetrix founded?
So it was like 2002, 2003 in that time frame, and I.
Mm-hmm.
And maybe a little earlier than that in some of the early technologies, but I joined 2006, 2007 in that time frame. So
right. So really,
kind of done the first peak of it become really famous and well known as the microtechnology became pretty well adopted.
Yeah. Well, so basically, that's you were with the company from its early days and then all the way to exit, right? So there was the acquisition by Thermo Fisher. Yeah. What was it that made Affymetrix so attractive to Thermo Fisher?
I think it fit right in their portfolio. You think about the development of different technologies, and you think about, and we used to talk about this a lot at Affy, is using the right tool for the right job, and as technologies go from being super innovative to more mature, your ability to use them in more routine applications becomes better. And so, as an example, the pandemic taught us that, doing a PCR test, for example, was not easily, but widely disseminatable. You can really get it to the ends of the earth to do testing. But, PCR technology is obviously very mature technology, as is microarrays. And so that microarray technology, I think the idea is that when it's, very stable and very well characterized, and you're really trying to look repetitively in a very inexpensive way at parts of the genome, that a microarray really does a good job of that. And that wasn't something necessarily that was in the Thermo life science group portfolio. There are obviously PCR applications, and then you had the sequencing applications that they had through their Ion Torrent acquisition. But there was this gap in the middle, and that's where that technology fit in, and really played a role in the middle. And I think that was the rationalization at the time.
Yeah. I mean, it's really interesting as we start to talk about, the DNA industry at large, how, nuanced it is when you start to get into the different applications. So like something as, I don't would say simple, because it's actually a very brilliant experiment that Kerry Mueller designed in one of, I think, his LSD trips while surfing. This idea that, for the folks listening, polymerase chain reaction, that's what PCR stands for. It's a way to basically amplify segments of DNA so they become, big enough so that our instruments can detect them, because, a single DNA molecule is, imperceptible, certainly by the human eye, but also by instruments at the time, although obviously that's improving. So the idea that you can make copies of small segments of DNA, it gets it up to a, reasonable concentration, so you can actually elucidate what the sequence in a specific targeted area looks like. And the way that was done is actually, piggybacking on the molecular machinery of bacteria that live at super high temperatures, because the way PCR works is you subject the DNA to multiple cycles of heating and cooling, because when you heat the DNA, it separates, and as it cools, these heat stable enzymes that you've borrowed from thermophilic bacteria kind of bind the DNA with specific primers that you've designed ahead of time, and you basically do cycles like that over and over again, and you exponentially increase the copy number of a particular sequence. So in the case of the COVID virus, the specific screen, there were conserved regions on the virus that you could design primers for, and then amplify the variable regions in between, and get a sense of the nature of the pathogen that folks were infected with. So that's PCR. And then you've got on the far end is like the whole genome sequence, a practice that, technology that for the first time that was applied to humans, let's just say it was $4 billion, right? And end to end. So now it's commercially available for less than 400, which is pretty amazing. But that's every single base pair, like all 3 billion plus base pairs in the genome. But then you've got this sweet spot, between PCR and whole genome sequence, which is the microarray. So maybe you don't need all 3 billion. You need more than, like, say, a 500 or 1,000 base pair sequence, which is like what we're talking about for the viruses. So maybe you need, 1,000 markers or 100,000 markers or 1,000,000 markers. You can, essentially, leverage this plate technology and processes referred to as hybridization to detect if specific single nucleotide polymorphisms are present. And so has application in agriculture, has application in, personalized genomics.
Yeah,
So
cytogenetics and cancer detection. Yeah, there's all kinds of applications. And actually, going back to what we were talking about, thinking about the international work that I did, one of the points that I remember making to Frank Whitney, who was the longtime CEO of Affymetrix, was, when you have a large population in a country that's still developing, you have a couple of needs that fit very nicely with what we do. And that is, you have a lot of research using genomics in agriculture to sustain the food supply. And then you have an opportunity to improve diagnostics. And you have an opportunity to use molecular techniques to improve the diagnostics and to get more precise in diagnostics. And that was one of the points that I made to him many years ago, is that as we grow as a company, focusing on some of those markets and thinking about the mix of population and investment in those could be good for commercialization of these technologies. And I think if you have a very US or Western Europe mindset, you forget that think about the population of the United States being about 330 million people. China and India are both 1.4 billion. And, obviously there's different access to technology around healthcare, and the healthcare is not as advanced, but there's still a big population. Southeast Asia as a group, you have, and Latin America both have over, 650 million people or so in each of those regions. In the Middle East, you have about 330 million. So that's equivalent to the US. So you have all these pockets of very large populations that have a need. And they have a need because their economies are advancing, the governments are investing, and they have a need to stabilize the food supply and improve on the use of molecular techniques into medicine and healthcare. Because typically when you use more advanced techniques, a lot of times it can be less expensive and you can get a more precise answer, which reduces the strain on the healthcare system. And so that kind of as a rationalization to Affymetrix at the time meant that we invested a pretty good amount in growing our international effort, and that paid off with having businesses in each of those places, in China and India and the Middle East and Latin America and Southeast Asia.
Yeah.
So it was I think actually a really good investment that spread genomic use in a lot of different places in the world.
Yeah. I think you guys even did some work with the VA, right? The Veterans Association back in the day in collaboration with the Kesogen. I think you were part of that 1 million veterans project. Isn't that right?
Yeah, we were the technology provider for both the Kesogen and then Rutgers.
Rutgers, yeah.
Had a core, and those two were the service providers, and we were the technology partner for the VA. And so yeah, it was, the idea initially, and there was a lot of, at the time, there was a lot of discussion about that. You mentioned whole genome sequencing, and the idea is, we have these million samples, or the other one was the UK Biobank. You have these large biobanks and large repositories of samples that get collected. And the natural tendency is we want to use the most advanced technology. We're going to try to do human genome sequencing on a million people. And that's a very admirable goal. But the other part of it is, to get to a real outcome, that's also a lot of data analysis. You think about the amount of sequences per person, and then you multiply that by 1 million. It becomes a very big haystack. And, trying to find significance of that. And that was the pitch basically that we made at Affymetrix to the VA, to the UK Biobank, is the idea is do sequencing and get a custom design that you can look at the most important single nucleotide polymorphisms that are important. What makes each person different? What are the things that are significant in the population, the SNPs that you want to look at, and put that on a chip, and then process 1 million people. And so that was a workflow that became very popular, and we did that for a lot of people beyond, the VA and UK Biobank were some of them, but there were a lot of biobanks around the world that we did the same thing. And it's interesting too, because you end up when you have these large genomic projects in different parts of the world, they become very important scientifically to the population, and because they're looking for the first time, sometimes very deeply into the genomes of a specific people, whether it's Saudi Arabia or Korea or Brazil or wherever. Looking at the genetic makeup of a country, I think, is really important. And it's interesting. One just quick story around that is one customer I had that was leading the genomics biobank project in a particular country. We convinced them that our technology can customize around the population of the sequencing that they were doing, And he was very successful with that project and became very well known in the scientific community, and was already very highly leveled in the country around science. And ultimately, I had heard about 10 years later, became the president of the country.
Huh.
And actually, the science tie-in and the respect for science in this particular country was high, and I think was able to end up becoming president of the country because of the scientific, in part because of the scientific accomplishments that person was able to do.
Amazing. Well, you mentioned UK Biobank. There's a VA project, and I mean today we have like all of us. So 10 million Americans, I think it is. What's the UK Biobank? Is that like a 500,000 or is it a million? I can't remember.
I don't remember. I want to say it's 500,000.
Yeah, I think you're right. But interestingly, that has given us this age of polygenic risk scores.
Mm-hmm.
To your point, is you've got like multiple SNPs, single nucleotide polymorphism, so points in the genome that are variable, conserved, and presumably elicit some kind of evolutionary effect on the population. So something measurable, that might be lactose intolerance, or that might be gluten intolerance. It might be a decreased ability to absorb vitamin D. There are all these kind of like minor enzymatic affinities that are associated with these base changes, and the
fast
idea,
metabolizer for a particular therapy or slow.
Precisely. So pharmacogenetics, right? Mm-hmm. Huge right now. Mm-hmm. Actually, anecdotally, I had a knee surgery last week, and I'm fortunate. I have my whole genome sequence, and I'm fortunate to have been thinking about DNA for 25 years. In a combination of both academic and industry, and so I have the latest in terms of technology as it relates to my DNA, and I had a pharmacogenetic report, and I have AI layered on top of it because the pharmacogenetic report is like 100 pages long, and the meeting with my anesthesiologist was going to happen in 5 minutes, so I didn't have time to analyze that report, but I did apply the AI to it and just say, hey, what, if anything, should I ask or tell my anesthesiologist? And it actually gave me like the top 3 things to tell in terms of pharmacogenetic impact as it might relate to what set of drugs he would choose to put me under. And then actually a part of my treatment plan afterwards was Celebrex. And as it turns out, my genetic profile makes me like at an extremely high risk of negative impact on Celebrex. And, those are so severe that, it might be, high blood pressure or stroke. And it was funny because this wasn't my first rodeo. It wasn't my first surgery. I've had a few injuries in my past as a result of, my athletic ignorance. Let's say I didn't have my DNA when I started, or at least I didn't have my DNA sequence when I started. So I've got all these injuries. I've had a few surgeries, and I remember getting Celebrex before. And at the time, I had my Oura ring, and I remember laying in bed, and my temperature is just skyrocketing. I have a resting heart rate of 120. When normally my resting heart rate's around 45 in the morning, and I'm just like, I had to get out. I remember going for like a 45-minute walk in the dead of winter because in my crutches, because I was just like, I couldn't calm down. So anyway, the point is, I passed that information on to my anesthesiologist, and my treatment plan changes, which is amazing. And the idea that that's just pharmacogenetics, but there's risk of cardiovascular disease, there's risk of Alzheimer's disease, there's risk of various cancers. And what we're learning more and more is, the more we understand our DNA, the more we understand the extent that these diseases have an aspect of heritability. So, certainly some diseases like Huntington's are, like 100% heritable. It's a complete genetic factor. But then there are others like multiple sclerosis, like what my mom had, which is both the genetic influence, environmental, and behavioral. So, but the thing is, your DNA, then you presumably, if you want to, and you're able to, you change your environment, and then you intentionally change your behavior to get the outcome that you want. So in the case of multiple sclerosis, it's, we know that those with the higher risk of multiple sclerosis, the further you are from the equator, higher risk of developing the progressive form. So the idea is, hey, if you can live closer to the equator, do it, and then limit inflammation. So that becomes, diet, exercise, being hugely proactive with that. And and then you can get into all kinds of other, precision diets and nutrition and supplements. But, all of that is something that we can start to, teach our next generation. I start to think about everything I'm hearing about longevity and living to 200. And it's like, I think we can do that if we start teaching, people when they're like, 10. Like, I would love it if by the time everybody's 18, they have like a PhD in their own body. I'm being a bit of an idealist there, but just kind of pulling the thread on these different applications like that you mentioned, like population genomics. What we were interested in Athletigen was like athlete genomics. So it's like, what is it that makes athletes superhuman, let's say, in terms of injury resilience, training response, and call it readiness based on their nutrition. I'm interested to know, what are some more of the more interesting projects that you worked on, either with genomics or some of the other technologies that you've deployed.
Yeah, I think one of the things that you're hitting on, and just to come back to that is, I think oftentimes physicians are a guide to people and how to understand their health and their healthcare. And one of the key things is really physician education. And I remember hearing a talk from a guy who isn't with us anymore, Mike Christman, who used to be the head of the Coriell Institute in Camden, New Jersey. And he did a number of talks for us at Affymetrix. But it really sticks with me. And that was, he was making the point, he was one of the world experts in pharmacogenomics. And he was making the point is that one of the biggest opportunities or needs is to educate physicians on the use of personalized medicine, on the use of using genomics in medicine, and how important it is. And, back to your specific question, one of the areas that I worked in was breast cancer. And, my mom passed away from breast cancer. And,
Sure.
So you really end up having a very significant connection to the, to doctors that work in that field, and really understanding the specifics about that. And one of the things that we focused on when I came to it was a company that did early stage breast cancer testing. And it wasn't, do you have breast cancer or not? It was based on your genomic profile, do you need to have chemo or not? And would chemo be beneficial or not? And it was a genomic gene expression profile that would give you the answer. And it was backed up by a very in-depth clinical trial that was prospective, not retrospective. So looking in the future and predicting, based on the test, compared to how someone clinically presented, whether or not a woman with early stage breast cancer would benefit from chemotherapy. And the biggest thing that we needed to take on was educating oncologists, educating physicians, educating breast surgeons who were going to be doing the operation about what the benefit was. Because, from what I came to learn and understood is that, going deeply into genomics is not at the forte, or not at focus of what a breast surgeon learns, or what an oncologist learns. And so being able to educate physicians about why a test is necessary, or why a test is necessary and could end up meaning that they don't need to give chemo to a patient, is an important thing. And I think that to me, if you're talking about really the dissemination, you get the way that I think one of the big ways that you disseminate the use of genomics is that you use the medical system, and you use doctors to be able to explain it, because that's what a lot of people in society go towards in many parts of the world, right? Is you go to the expert, you go to the expert of and it's, that should know, has been educated and has spent a lot of time being educated around how do you apply these things. And I think one of the things that's interesting is that genetic counseling is becoming a huge area of opportunity and expertise that's being needed. And it's interesting. So my older daughter, I have two daughters. My older daughter is a senior in college, and actually she's interested in trying to become a genetic counselor. And I think some of it's related to just being around me and hearing all the different things around genomics around the world, and the use of genomics and integrating it into life. But I think it's important that there's a group that can simplify it, demystify it, explain it. You actually gave a very succinct explanation. You're an expert, but you turned to AI right before you talked to your anesthesiologist about how do I boil this down into 3 points. And this is an expert talking to an expert. And so you think about someone who's not as informed in genomics as you, and how do you really get to have that informed conversation with your physician, because it impacts all of us, right? And we're all made up of DNA, and the technologies are becoming more and more accurate. They're becoming the ability to look into the genome and have real medical world implications is becoming more of a reality. When they first sequenced the human genome, now 24-ish years ago, finished it. There was thinking that this was going to happen immediately, and I think part of the missing link is that it is educating those people where people get their help from, and I think that that's still a big need.
Yeah, it's funny. Just to pull on that thread, you said, okay, I'm an expert talking to an expert, and we have a translator, AI, right? So it's like, I know DNA, the anesthesiologist knows drugs. Between the two of us with AI, we can start talking the same language.
Right.
But it's interesting, like, again, the need for that's high tech, the need for the high touch. And I'll give you an example, like the high touch. What demands high touch, for example, is, when my wife and I had our daughter a year ago now, and about, a month, 6 weeks, 2 months in, Brooke was, failing to thrive. Like, she was not gaining weight. When we went in, she was first in the 12th percentile. And we're like, oh, that's okay. It's not a linear thing, and that's how it goes. But then it's like, okay, well, now she's the 5th percentile, and now she's the 1st percentile. So like, what's going on? And, we had her DNA sequence. So I had my genome, I had Kristen's genome, and we had Brooke's genome. And so I didn't really have any, well, that's not true. I had dived in initially proactively to, kind of determine or rule out some things that I knew, I was a carrier for, Kristen was a carrier for, et cetera. But, then we had this failure to thrive thing, nutritional issue, and, there's urgency now. And, we were recently dealing with a death in the family. So there's all of that emotional load that goes on in parallel to that. And so, that whole idea of being like emotionally compromised, it's hard for everybody to kind of be their own physician or be their own advisor. So yeah, the need to like get out of the echo chamber and have a conversation with somebody who can help you, develop your plan, an advocate for your health, so to speak, and understands the technical and the more nuanced, but also like the emotional element. So yeah, coaching and counseling and all those things, I think are going to become more more important, where it's not so much the answer that we're interested. Of course, we're interested in the answer. We're interested in, the information that's going to drive the outcome that we want. But it's not apparent how to get to the answer. Well, it is. You have to ask the right question. So how do you ask the right question, right? Because when the AI initially spit something out to me that was unintelligible, I knew how to correct the prompt and ask properly, and et cetera, et cetera. So, there's this day and age that we're entering into, I think, with data, any data at our fingertips. I truly believe that, what are we, 2024? I don't know if it's aggressive to think that everybody's going to be sequenced by 2030. Maybe not everybody will be sequenced, but maybe we'll be at a point where at a certain point, genetic sequencing is at least offered to everybody by 2030. The costs are plummeting. Whole genome sequencing is like, costs are let's say under 400. That's just only about going to become more efficient as we remove humans from the sequencing process, better standardization and tests. But then, of course, it's the data storage and the analysis, right? So,
well, I think that's the key, right? Is the analysis and the interpretation, and
right.
and to what end, right? And I think there's some obvious ones. There's some obvious applications. So pharmacogenomics is the one that is super super obvious. That, but I also think about one of the other things that I think is still quite an interesting space is thinking about the microbiome.
Yeah.
Thinking about everything that you put into your digestive system has such an impact. And you were talking about risk and thinking about what you ingest and what you eat is such a component of your health. And I think looking at that and looking at the sequencing related to that is I think really important too. But I think it's got to come down to the applications. I mean, the technology is becoming more accessible to open up the capability, but you have the layperson who may they need a reason to get their DNA sequenced. They need a driver. They need
right.
It's got to be for a specific end. It's got to be for a specific use case where they're gonna need it for something.
Yeah. Well, I think the more, like, they call it the motivated groups that I've come across as of today are at like two ends of the extreme. Okay. You've got rare disease, right?
Mm-hmm.
The rare undiagnosed. And then you've got elite athletes. Both of them are looking for that like 0.1% variation. Like, what makes me different, right? Now, in the case of the rare disease individual, it's like, it's a life or death scenario. They're on a diagnostic odyssey. Some of them have been on this for like 10 years struggle. And like the outcomes that I've seen from whole genome sequencing being applied to that. Now, to be clear, these aren't existing panels, right? These are people that know enough about their biology to be dangerous, and they go in with some prior knowledge. They've built a network of experts, of others in the community who have been suffering, and they've identified specific markers or specific sets of markers associated with their conditions. And frankly, it is life or death as far as they're concerned. From the athlete's point of view, it's not physical life and death, but it's psychological life and death, right? They identify as an athlete, and the difference between a gold medal and not making the final is
fractional.
Yeah. So, knowing where the quicksand is in terms of like injury predisposition. Like I spoke to that before, right? I spoke to, I've had 7 surgeries as a result of, a very simple heuristic that I could have applied had I known my DNA earlier, which is, so I look at it across 3 areas. There's the structural integrity of your body. There's a physiological and structural response to specific stress, strategic stress, that is training. And then there's your nutrition and metabolism. So that is, how it is you fuel and recover and rejuvenate. And what I've learned from my DNA is 3 unfortunate things is, well, unfortunate or fortunate, it is what it is. I got a monster truck engine on a Chevette chassis, so everything breaks, and I burn dirty, right? So I there's foods that I'm intolerant to that lead to autoimmune responses. And when you're younger, you can get away with that, but if you're pushing the edge, your inability to recover or your risk of injury is going to reveal itself sooner than in others. So I mean, I think it really is. To me, those are the two most motivated populations.
Well, and I would actually add a third and introduce one of the technologies and projects I've worked on over time, and that is cancer patients. And,
Sure.
and thinking about the tumor microenvironment, and it's not DNA, but it's RNA, and thinking about, so I've worked at Resolve. I was the first commercial leader there, and then I actually do some work with another spatial company called Curio Bioscience. But the idea is, one of the things that I think has still been missing is the spatial context, especially related to RNA, and especially RNA and protein, and thinking about it, in a tumor microenvironment. I mean, you can apply it a bit as well to things like traumatic injury or CTE or those kinds of things. But where you really have a big applicability is looking at a tumor and looking at because tumors are very heterogeneous. They're not they're very complicated things, and looking at at that spatially versus looking at that looking at how closely is something connected to something else, and that is, I think, another level of complication, but another level of potential discovery where you can understand that if something is closely located, then that has significance. And I think that's another technology that's really becoming interesting and becoming more widely used. There's a lot of companies are investing a lot of money in that space, and it's really the evolution. Back to thinking about the evolution of technologies and tools on the RNA side rather than DNA, but thinking about the Affymetrix microarray and then RNA-seq and then single cell sequencing, and then spatial is really the next thing beyond that. And I think that has the potential, especially in oncology, to really help a lot of people and come up with very specific use cases. As an example, should I use this drug, this therapy, or that therapy? Because you have specific use cases where you can look at the spatial context and understand that. And I think that, to me, is kind of some of the stuff that's coming that has the ability to be super interesting and super informative and very, very helpful. And where I think the use of genomic technology has the ability to get into healthcare pretty quickly, because the impact has the opportunity to be massive.
Mm-hmm. Yeah. So tell me about the work you're doing with Backstitch DNA. What's going on there?
So it's it's a new technology that is from Stanford University. Is a company that's spun out of Stanford, and it's just very early stage technology. And the basic idea is being able to stitch together, that's the name Backstitch, stitch together long pieces of DNA. And so the idea is that can be used in various applications in ag and metabolic engineering, and thinking about the development of new therapies, thinking about the development of new constructs for DNA, and synthetically stitching together pieces of DNA that could be then consumed. And so it's like I said, it's new technology that is just in the early stages of being validated externally to the company, and we're looking for collaborators that have interest in trying it out and seeing what they can do with that. Because at the end of the day, a lot of these technologies, companies innovate, they make it, and then they put it out into the community, and then it's what can be done with it. And I often use the analogy of a smartphone or a computer. You have the platform and you have the system, and then you have applications, and the applications are really where you get the value of what having a device. And I think the same analogy holds for genomic technologies. And you have these platforms, but really it's what do you do with it, and what does it enable you to do is really where the payoff is. But yeah, that's a little bit about vaccines.
I like the analogy. I want to just press on that, but I want to come back, make sure I understand the technology you're working on a bit better. But
Sure.
the analogy of like you said, a smartphone, and even back up before that, I remember from one of the Jobs movies where I think they're launching one of the first computers, and the whole vision, Jobs' vision, is that everybody is going to have their own personal computer, and it's just like, well, what are they even going to do with it? And then I think it's his daughter is there just kind of playing and draws a picture and prints it out, and
Right.
then he holds it up and he says, this is what they're going to do with it, right? And it's funny because you look at it, it's like, well, what is that? And it's like, well, that's a human expression, isn't it? Like through technology.
Creativity.
Yeah, it's creativity. And well, interestingly, I think one of their first markets was academics. And so yeah, the application, I think they had a lot of application in education and in design, etc. And now everybody has one in their pocket, and the applications are limitless. And that's kind of daunting, right? Like that's a challenge as somebody who commercializes new products that are game changing, that are effectively limitless, like such as DNA. It's like, well, everybody's your customer. Anybody who has DNA is your customer. So as far as I can tell, that's everybody. But you're no farther ahead, right? Because you're like, well, what application, what use case? And so then actually maybe we can finish with that. Like, I want to understand your process a little bit in terms of like, how do you choose and prioritize the application? Because it seems to me there must be some fundamentals or first principles that you run through in terms of economics, supply, demand, markets who would be best served by the tech,
Sure.
which is probably not always apparent. So, but backstage, I'm going to just try to understand it based on the name. I'm probably going to get it wrong, but so I'm thinking it's large constructs of DNA that are put together strategically to form, like, are they bacterial operons, or can they be put in animal and plant cells for gene? Like, are we talking about GMOs, by the way? Like what are we talking?
Well, so the technology is generic in that it stitches DNA together, and so it's really then again what the applications that could be used. So sure, you could have someone in ag bio that wants to do something synthetically. You could also do that for other applications. But so there's a wide range of the utility of it. It's really an enablement technology.
Got it. Do you have like any initial applications that you're?
Well, certainly ag is one of them. But protein engineering, looking at metabolic engineering, looking at the the focus of who's interested in having really long constructs of DNA that are customizable.
Got it. Okay, so I'd love to maybe finish just learning more about your process to commercialization, and maybe we can put like a future slant on that, because it would kind of foreshadow maybe where you think things are going. So you get a new tech, like for example, backstitch DNA. What's your process for choosing a market? Because you do, going back to like the spirit of the call initially, it was like you come in and you come in with a different perspective, a critical perspective, like having been, I call myself a recovering academic, staring down the microscope or into the molecules too deeply, and your blinders are up. For all intents and purposes, can be like that ostrich with his head in the sand, not deliberately hiding, but interested in the ground, what's going on there. And then you come in with this perspective. And so, how do you, what's your process to the analysis? And then there's got to be like the socialization and selling the compelling future to the people you're advising. So, what's that process like?
Yeah. So, to me, it always starts with who's going to use this innovation and why, and what problem is it going to solve? And do they recognize the problem or do they not recognize the problem? So, I think it's really thinking about that, because ultimately, you've got to get an understanding of who's your target, who could potentially use it, and how many people like that in the world are there? Is there 10? Is there 1,000? Is there 100,000? And do they have the ability with this innovation to pay for it? Do they have the ability to use it in their work? Do they recognize that this is going to be something that's going to be beneficial to them, or do they see it as a threat? So I think you have to evaluate all those things, and as part of the business planning process that I help a lot of companies with, is to thinking about those things and trying to come up with the answers to those questions, and to think about, what problem are we solving? Who are we solving it for? How many people are like that? And how well understood and characterized is that problem today? Is it something that you really have to do a lot of education? Because that takes a lot of investment to educate people about the need. Is it something that is obvious? Is it, I see a lot of examples where people will work around the issues that are in existence, and they don't recognize the problem because they have worked around the way that they work to avoid dealing with this problem that, if you fixed it, would make their life so much easier. And again, I go back to a lot of the the analogy of the smartphone. You think about, an application like ride sharing applications, right? I mean, did anyone know that trying to hail a taxi was an issue that could be enabled by a smartphone? And ultimately, the answer is yeah, it has become a great innovation. And there's a couple of companies that have become very large, successful companies by understanding what the problem that they're solving is, who they're solving it for, and understanding that it's a very large market. And so it's really thinking about that from a customer's vision, a customer's perspective. What are you solving? How are you solving it? And how easy or difficult it is to demonstrate the resolution of that issue. And are people going to give you credit for that or not?
Got it. How right are you the first time?
Almost never. It's
Yeah.
you create, based on your experience, a set of assumptions, and you work on those assumptions, and then you refine them, and then you have it based on experience. You have an idea where to go, who to go to, why they'd be interested. But part of it is for figuring out what's the message. How are you going to talk about the innovation? And there's a refinement. There's a honing. There's an evolution of that, and that's that process takes time, and that's a commercial process that takes time. And it's interesting. I remember meeting an innovator that had developed a company that he was in the process of selling, and his comment to me, because we met through him selling his company, and his comment to me was, if I had met you, someone like you, when I first started this process of creating my company, I think I would be a lot more successful. Because he was an innovator that had an innovation that was really super interested in having his innovation become famous and well used. But what he didn't get was feedback from the market to help him innovate, refine, and message correctly about how to get that innovation widely deployed.
Got it. Yeah, it's like those minor adjustments at the beginning of company design that have, exponentially can add exponential value or lead to catastrophic outcomes.
Yeah, I mean, you look at a lot of the big companies in our industry, Illumina, 10X, I can think of off the top of my head. The thing that they became famous for wasn't their first innovation.
Right.
And I think, part of it is becoming a commercial company and having a product roadmap and having an understanding of how a new innovation would fit in the market. And in some cases, it's acquired because the technology is not resident in the company, and in some cases, it's innovated, and then it becomes a home run in the case of next generation sequencing and single cell sequencing.
Yeah, amazing. So, what's got you like most excited about the future? Like, you've been in this field for decades. You've seen where it's come from. Where is it going, and what are you betting on right now?
I think the technology innovation it is one, and I think that there's been a little bit of a lull from a technology innovation a bit over the last couple of years. I think there was a bit of a hangover uh, from a there's a lot of money and a lot of innovation in the pandemic, and then a lot of that dried up and changed, and then there's been a couple of years where investors and new companies have really, it's been a lot harder to get on the other side of that innovation. But I think that is going to pick up in the next year or two. And I think that advancement will continue. And, and I think about whether it's, as I mentioned, spatial, synthetic biology is another area. I think those areas are super interesting. But to me, from a personal perspective, I would, use the overused word of democratization, but it's really getting out to the ends of the earth. The use of genomic technologies. I mean, COVID showed us that you can put a PCR machine in the edge of, the corners of the earth and do reliable PCR testing to understand whether or not someone has COVID or not, and deploy therapy against that or a vaccine against that to minimize the amount of spread. And I think that I've heard a lot of subsequent to the pandemic, a lot of people talk about, we want to do the same thing for different kinds of cancer or whatever it is. And I think that those are very great aspirations. But I think that, to me, that it's a big world, right? There's over 7 billion people on Earth. And, if you look at Western Europe and the US, where a lot of this technology is innovated from, there's still a lot of places to go to apply these technologies. And I think that to me, and the application of that is the thing that I get the most excited about based on the work that I did at Affymetrix. I think that is interesting to be able to apply it, because that is potentially economy changing to a country. That's potentially very helpful to a healthcare system for a given country. And, I think any country that decides to invest disproportionately in that kind of science has shown over time that they do quite well. And, I think that there's good examples of that in lots of different places.
Gotcha. Okay, Chris, at the top of the call, we talked about some rapid fire questions. You ready to go through those?
Sure.
All right. So, right. I think we've been saying it without saying it, but let's say it now explicitly. So, what's the key message here that we're passing on to listeners? We've been saying it, but maybe we've been saying it without saying it. What's the key message that that you'd like to share based on your industry and personal experience as it relates to genomics?
Yeah, I think it's a little bit of what I just mentioned. It's a big world, and there's a lot of places where we can apply this technology. And this is just the tip of the iceberg. This is just the start.
Right.
You think about, as I travel around the world, the ubiquitousness of smartphones. I think that the same opportunity exists, for DNA technology around the world. And the amount of usage is still very small.
Yeah. And so that leads or begs the next question is like, well, who's the one person or group of people that needs to hear this message to amplify that application?
Yeah, I think it's companies and how I think that's one constituency is companies and how they prioritize and certainly prioritizing and focusing on investing in the US and Europe. is always important because that's where the biggest usage of these technologies are. But don't forget the other countries, and don't forget the opportunities that lie outside of those two main markets. I think that's one, and then I think governments, their relative influence in that, and then the last group would be investors. Pushing companies to say, let's take the next step and go into markets in Asia, go into markets in Latin America, in the Middle East, Eastern Europe, and so forth, because there are a lot of people with need.
And then the last question is, who has to be on the DNA of things? Who would you nominate?
I think the thing that's super interesting is kind of following the money and where the early stage investors, how they decide, why they decide to fund different technologies. And so my suggestion would be those in the venture world. And there's a lot of different venture companies that have also created. I mean, I'm a part of one around doing executive residence at General Inception. So, Paul Conley is the head of General Inception. He would be a good person. I think Illumina Ventures, Nick, who's the founding partner, would be an interesting one. I think they have a huge amount of influence about what companies become real and grow, and I think that decision making process is super interesting.
Awesome. Okay, so that was Paul Connolly, and sorry, what was the second name from Illumina Ventures?
Nick Niclario.
Nick Niclario, right on. Awesome.
Yeah,
all right, well, maybe I'll seek up.
Co-founder of Illumina Ventures.
Awesome. Okay, and finally, Chris, how can people best find you, get in touch with you? What's the best way for them to reach out?
Probably the easiest way is on LinkedIn, and just send me a message through LinkedIn. I'm pretty active on that, and that's probably the easiest way.
Perfect. We'll put that in the show notes. Chris Barbazette, thank you so much for being my guest on DNA Things.
Absolutely.
Well, thank you, everybody, for joining us today. What an insightful journey into the DNA of things. Today's conversation with Chris Barbazette shed light on how bridging technology, science, and commercialization can impact health outcomes worldwide. Now, Chris's unique approach to expanding biotechnology access offers a compelling vision of healthcare that reaches beyond borders. Now, we hope you've gained valuable insights on how understanding and applying genetic data can shape not only individual health journeys, but also global healthcare. Now, if you find this episode inspiring, please share it with others who might benefit. And let's continue to spread the love for science and unlocking the potential within our DNA environment, and making those behavioral changes to activate our human potential. So thank you all very much for listening. One final special announcement. As some of you listeners know, I work exclusively with those folks I like to call creators or change makers. And my clients tend to be executives, athletes, entrepreneurs, artists, the creators of tomorrow. So if you are, or you know somebody who is looking for some insight into their DNA and how to unlock it, please feel free to check out my website at drjeremykoenig.com, sign up to the newsletter, and I'll be reaching out with more details in the coming weeks. All right. Have a great week.